And thank you for coming, because I mean, it's the last day, so few aficionados are still here trying to understand what I can speak about this problem. So what I want to say today is a kind of short review of the project that is going on since about 3 years. These are the people involved in the project. Platab is working with Federico in Eindhoven, David is in Harvard, Mogens, as you know, is in Copenhagen, and now Simone got a position in Barcelona. So almost, this is almost a European project which has not been financed by the European Community.
Greece is missing.
Huh?
Greece is missing.
This is missing.
It's okay, everyone else is paying.
So what is the subject of our interest? Right now, by the way, we are all excited by new experimental results that are taken in Eindhoven, but unfortunately I will not be able to speak about that. But let me try to focus on the problem and see what we have been able to obtain in these 2-3 years. So the basic idea is that what we want to study is how the population of bacteria, individuals, whatever you want to call it, can spread and eventually change, mutate, grow, and die in a medium. Now, the basic approach for this problem starts from the Fisher equation. The Fisher equation is a very simple one. I mean, it's a reaction diffusion equation where you have a growing rate for the population concentration to grow, and you have a kind of cutoff. That is to say, the population can't grow more than some limiting value, which conventionally is taken to be 1 in the system. Now, this equation can be solved even analytically, at least in 1 dimension. And what you discover is that if you start with the kind of peak in 1 dimension, then you have a nonlinear wave which is propagating with the velocity which is, for dimensional reason, proportional to the diffusivity and the growth rate, and with the thickness of this wave, which for the same reason is proportional to the diffusivity divided by the growth rate. So this is more or less correct when you look at the spreading of bacteria on a Hagar plate. Okay, so in a very simple situation where the amount of food, if you want to...
Inverse of this, no?
Sorry?
It's the inverse. These... No,
this is the velocity.
This is fine.
Okay? So...
No, it's fine.
Okay. Now, the most interesting point comes from the fact that most of the biological interesting features in these problems comes into this region, where you have a decrease in the population concentration, and then there is an increase of fixation in mutations and all other interesting behavior in biology. But for the time being, let us concentrate to the problem that what happens to this equation when we have a turbulent flow. Now, there are 2 main classes of turbulent flow that you may wish to consider. The first one is incompressible turbulent, which should give you something like an increase of diffusivity. Now this is expected, and more or less this is been observed in numerical simulation. The other possibility is that the flow is not compressible. I mean, is compressible, and therefore you may have a different behavior of the concentration because of compressibility effect. Now, how and why one should consider the case of compressible turbulence? Now, if you think of bacteria in the ocean, or phytoplankton, so to speak, the compressibility can arise for different reasons. Bacteria have buoyancy, so they can stay on a given surface in the water, even if the flow is three-dimensional, and this is one case that we will discuss later. There is a large-scale compressibility due to mesoscale frontogenesis and all these other problems. Then you have a small difference in density between bacteria and the water, which can give rise to inertial effect in the bacteria itself. So there are all these effects that from time to time can be interesting and important, and therefore it is reasonable to introduce a compressibility effect to understand whether compressibility can change or not the behavior of the concentration of bacteria. Now, for the case that I will discuss today, compressibility has been introduced in a very simple way. We take a surface flow, like the one considered by Buffet and others, and then we look at the behavior of the population on a slide of this compressible flow. Because in two dimensions, if you take the three-dimensional flow and you cut it in two dimensions, then the flow in two dimensions becomes compressible. Now, this is a snapshot of a numerical simulation that has been done recently, and I will now show you the movie to get you a feeling of what's going on. And this is a snapshot of a recent observation done by satellite. The interesting point here is that they were being able to do two things. First of all, to classify different species in the phytoplankton, and second, to make correlation between the different species and the shear flow. And they found that there is a strong correlation between the velocity field, the shear velocity, and the different species with segregation effect, which are closely related I mean, this is a kind of Lyapunov estimate, but you can take it as a measure of the shear. So there is something that is reminding us that probably in the ocean, compressibility is acting in a way that we still have to understand, but is certainly important to make an effort in making a simple numerical experiment and eventually experiment in laboratory to discover which are the effects which are important in population dynamics when you have such possibility of compressibility. Now, just to clarify my introduction, let me show you this movie, which usually I think is interesting. Now, on this part of the system, you have a standard without flow Fisher waves, which you will see that will grow uniformly in this domain. On the other side, with the same initial condition, you will have the behavior of the population with the turbulent flow, with the compressible turbulent flow. And then you can see, while the Fisher wave is uniformly expanding, the other behavior is characterized by very strong concentration of the population in very narrow streets. So kind of, let's say, quasi-localization of the concentration due to compressibility. Okay? So this is a picture of the phenomena that we want to investigate. Now,
in this figure you show from the satellite data, how do they determine the species? Is it color, is it ocean color?
well, what they have a new set of sensors, I mean, this is the point, by which they can distinguish the different temperature and eventual mass. And they have also local observation. It's a lot of work to do this kind of Okay. So another thing which is important for this problem is to understand that the CO2 exchange between atmosphere and ocean is mediated by phytoplankton. So that the amount of concentration is reduced because of localization, and it means that you are reducing the heat flux, the CO2 flux between the ocean and the atmosphere. So it's non-trivial to know how the full population is behaving in time. Okay, so let us start with a very simple case in order to understand which are the numbers that we have to consider in order to build up a simple theory. So let us consider a simple case of one-dimensional sink. I mean, so this is a sink in one dimension. Okay? So the flow is going in a single point. So it's the most simple compressible flow that you can imagine. And let us assume for the time being that you have no growing, so there is only Lagrangian particle. So essentially the Lagrangian particle will follow the trajectory that is the solution of a very simple stochastic differential equation. And you immediately understand that there are two characteristic scales into the system. One, which is introduced by the turbulence. I mean, suppose that gamma is not only a number, but is a characteristic number of the turbulent gradient. So you have a characteristic scale which comes from the turbulence. And the other one, which is the localization length, which comes from this diffusion and the gradient. Now, when the localization length becomes smaller than the localization due to turbulence, what we think is that the effect of compressibility, I mean this effect here, becomes relevant into the behavior of the system. So we have a first kind of inequality which tells us that when the diffusivity is relatively small, or when this product anyway is relatively small, or if you think the turbulence is relatively large, then the effect of compressibility becomes important with respect to simple localization due to turbulence.
V star is defined how?
Is the kinetic energy of...
But it's not in the first equation.
Sorry?
It's not in the first
So
equation.
it doesn't appear, so...
Where is it?
Of course it's not. I mean,
So
if
this
you
is
have
coupled.
turbulence,
No, so
if
this,
you have turbulence,
Yeah.
and you think that this is a characteristic gradient of one sink,
yes.
okay, and you have a turbulence with some velocity,
Yeah, but the question is how the velocity appears. Do you add it to the v to grad x or what?
gamma is the gradient of the velocity,
Yeah,
So
but
I
where is
see.
G?
okay?
So it's a gradient v gamma or
Right.
what?
Yeah. The gradient, gamma is the gradient of the velocity. Now suppose that you have a sink in a turbulent flow.
So I have to think about gamma as gradient of v.
Yeah, that's it.
So I still have a problem of dimensions.
So this is the case. Now, if you do the same game with the growing rate, which is different from zero, then you have the competition between the velocity of the Fisher wave, which is trying to expand the population, and the velocity of the sink that is trying to contract the population. So this gives you another scale which depends on the Fisher wave velocity and the gradient of the velocity of the compressible flow. And when this quantity, which is determined essentially by the gradient of the velocity, becomes smaller than the thickness of the Fisher wave, then you understand that the effect of compressibility overcomes the effect of the deterministic equation due to Fisher. And so you have an inequality which is telling you that when the gradient becomes larger than the growing rate of the Fisher, then you expect that the compressibility becomes important. Now this is crucial because at this stage you should understand whether such an approach is relevant or not for the ocean, because we know more or less what is mu for the bacteria in the ocean, which goes from 20 minutes up to 12 hours, and we know what is gamma in the ocean, which can go from few seconds if you think of very strong turbulence, or it can reach up to, let's say, 6 hours if you take frontogenesis in some cases. So you see that this inequality may be, I'm not saying that it is important, but may be relevant for the behavior of phytoplankton in the ocean. Now, just to make things a little bit more general, if you consider now the probability distribution of the Lagrangian particle in a turbulent flow with compressibility effect, which can be parameterized by this number, then what you can do easily is to multiply this quantity by p, make the average, consider the statistically stationary situation, and make the standard approximation that this quantity can be parameterized by the fluctuation of the probability distribution multiplied by the divergence square. And then you see that you can get an estimate of the localization length for mu equal to 0, which is proportional to the diffusivity, as we know. Then there is the effect of the square root of mu and square root of energy dissipation, which is the gradient. And then here it appears the compressibility. So when the compressibility starts to increase, the effect of this length becomes, the length becomes small. And this is exactly what I showed you before in a very simple situation. So this is why you have the effect of this star, the gradient, all the things that I was introducing a few slides before. So this is what we can think as a first, let's say, guess. Now, let us now consider the one-dimensional case. So let us move step by step from one dimension to two dimension, and then first take the one-dimensional case. Now suppose that you have a population which is here at the initial time, and then this is the time, and this is the space, and the Fisher wave is essentially a nonlinear wave, as I said, which eventually cover all the wave. Okay? With the turbulence that here is simulated by a simple shell model, what you observe that the Fisher wave doesn't develop, and what you have is a localization effect, which is maintained for a very long time. So this means that when you have turbulence, you go very close to a sink, and then eventually you follow the sink as long as the sink is there, up to strong intermittency. Now, this is a qualitative observation, but what is interesting is not the is the fact that the amount of bacteria, I mean the carrying capacity, how much bacteria you have in the system, okay, is completely different in this case in that. I mean, if you compute it, you see that there is a factor of 10 between the amount of bacteria in this case and then the one observed with the Fisher equation. So the first thing that we would like to understand is to make a prediction about how much is reduced the carrying capacity as a function of the turbulent flow. So the very first kind of prediction that we would like to understand is given the turbulent flow, can we say something on how much bacteria you will find in the system? Of course, in the case of non-incompressible flow, the situation doesn't change. The amount of bacteria will stay exactly the same as in the Fisher wave.
But the constrained to the strip is of the same order as the Fisher solution?
You mean how much?
If I don't average over the whole system, I just constrain the average to the strip.
No, it's bigger.
It's even bigger.
Right, it's even bigger. So if you look here, it's almost a factor. I mean, here is about 2 and here is about 1. So if I just
it's only the
look at
localization that is
no,
reducing.
it's a normalization. Yeah.
Localization. Yeah.
Localization, which takes you, okay. But if you are interested in the facts of...
So locally it will be larger.
Locally it can be larger. But this is exactly what you observe in the ocean. You have a concentration of bacteria in some places. So, the answer to what is the amount of carrying capacity as a function of the growing rate and the turbulence can be understood in the limit where the growing rate goes to zero. Let me try to explain how we can make this prediction. Suppose that we can compute the concentration, the probability distribution of a Lagrangian particle, essentially, for mu equal to zero. Okay? Suppose that we can do that. And let us assume that in this limit, when mu is different from zero, the only effect of the flow is just to make a rescaling of this probability. That is to say, the limit is singular because when mu goes to zero, you are always constrained to a different equation, because if I make an average of this equation in space, this term is zero, this term becomes zero, this becomes zero, so you have a constraint. Okay? But the probability to find bacteria in some position, the concentration in some position, is nothing else, nothing more than the probability of the Lagrangian particle, just multiplied by a number. This is a nonsense. We are not able to prove it, but it seems to be reasonable. And if you use this information inside the constraint, you immediately get that the amount of carrying capacity is nothing else than one of the square of the probability distribution for the Lagrangian particle. And this is the estimate that you get from the numerical simulation, and these are the numerical results that we get for different values of mu. And you see clearly that when mu is large, you recover the case theta equal to 1, and when mu becomes 0, you go to the limit which is predicted by this constraint. Okay? So essentially this implies that if you have information on the Lagrangian particle, then you can make a prediction on the behavior of the carrying capacity when mu is very small. Now this is true also in 2 dimensions, because if you do the numerical simulation in 2 dimensions and you look at the limit of very small mu, now this is mu multiplied the Kolmogorov time, so now the limit is done in dimensionless unit. And these are 2 different resolutions. I guess this is 128, 512. Then when mu goes to 0, then you reach a saturation which can be predicted by this expression that I showed you in the previous slides. So essentially the Lagrangian behavior is fixing the asymptotic behavior of the carrying capacity in the limit of very small growing. Of course, you can also make another step, and you can look at the multifractal behavior of this quantity for different values of mu, and then you can relate this quantity, which is, again, the fluctuation of the probability distribution of the Lagrangian particle, to the localization length and to the scaling exponent of the second order moment of this quantity, which, by the way, is scaling very well without any possible tricks. And so the final result is that the total carrying capacity depends on the localization length, which again is expressed in terms of diffusivity and all the other stuff, and this quantity, which is characteristic of the Lagrangian trajectory in the compressibility flow. So we have a prediction about the carrying capacity as a function of this localization length and as a function of all the other parameters appearing in the system, which, as you can see, seems to work rather well. Now, since you have a two-dimensional flow, which is compressible, you can always project the two-dimensional flow in the incompressible part and a compressible part. Okay? And then you can mix with different angles such that you can change the compressibility. This has been done also by Boffetta and others, and is done again in this case. Now, so you can study how much the carrying capacity change as a function of the compressibility. And what you observe is that even if the compressibility is relatively very small, I mean it's 10 to the minus 2 in this case, you still have an effect on the carrying capacity, which is order 20%, which is not so small. I mean, if you consider the 20% change in the carrying capacity means 20% change in the total population, and therefore 20% change in the CO2 flux between ocean and the atmosphere. Now, these are the results for different compressibility. I mean, this is compressibility equals to 1, so it means that there is only potential flow, and this is all the other intermediate case. You can generalize the analysis that I show you for the case of the carrying capacity by trying to parameterize the multifractal exponent as a function of the growing rate, the compressibility, and of course of the Kolmogorov time. Now, for dimensional reason, this quantity must be a function of this product and the compressibility, and since the characteristic time for the compressibility is related to the Kolmogorov in this time scale in this way, a reasonable guess for this number is something of the order of the square root of k divided by mu tau plus eventually a constant. And if you look at the numerical results, you see that more or less this guess is captured by the simulation. The exponent, instead of being 1/2, is ordered 0.62. But essentially, you see that this behavior is observed in the numerical simulation as for different value of the compressibility. Now, if you go on and you look at how the carrying capacity depends on this exponent, then you find out that essentially it is independent on the compressibility, and you have a very nice scaling law for different value of mu, and which is on the same line. And therefore you are able, by looking at these results, to show that all these different curves can collapse in universal, so to speak, behavior by looking at this simple and non-trivial combination of the different effects. So this is, again, a non-trivial result, because it means that changing the value of the turbulence, changing the value of the compressibility, what you are doing is just to move in this, on this line. As you see, universal is bipolar, because it's a particular case that we have analyzed. It is not clear whether if you put inertial particle or if you put other kind of turbulence flow, you will get the same results. But our feeling is that more or less the behavior of the carrying capacity as a function of the fundamental parameters will stay the same. Now, this is the first part of the problem. The second part is much more interesting, because now what we want to study is how the system changes when you have competition and eventually mutation, and on the top of it you have the effect of turbulence. So I will now try to summarize in 2 slides the huge amount of information. So please, I will skip all the details. So the starting point is to assume that you have 2 populations, that you have the same birth process with the same rate, and the same death process. So the 2 populations are neutral. You have red and green that do not care about each other. The only competition is on food. Okay? Now, once you know which are the rates for death and birth, you can write the master equation, and from the master equation, with suitable normalization, in the case you have only a single box, you arrive to a set of stochastic differential equations which describes the behavior of the concentration in one single box. If you had the diffusivity and the flow, what you have is a stochastic differential equation now in one or more dimensions with this multiplicative term. These are called, without the velocity, are called the Fisher-Kolmogorov, Piskunov, somebody else that I never remember, equation.
Petrovsky.
Huh?
Petrovsky.
Petrovsky, right. Equation. And these are playing an important role in many fields of physics. In particular, for the case in which the total number of population is fixed, the model is called the stepping stone model, and is an important model in trying to figure out what happens in the behavior of the birth and death process of bacteria in the experiment. And this is a rather counterintuitive example that you can explain by the stepping model. You start here in the middle with a well-mixed population. So essentially the red and the green are 50-50, completely well-mixed. And then you let the time go. And what you observe is the system naturally produce segregation. So the red and the green segregate, okay? And then the segregation happens, according to the model, the stepping stone model, on a time scale which is proportional to the square of the number of animals. So it's a rather relatively long time scale. Eventually, if the size of the system is finite, one of the two populations will be alive and the other one will die on a time scale which is called the global fixation time, which is order of the size of the system squared divided by the diffusivity. If you perform a numerical simulation starting with the same condition, then you observe that the model indeed reproduces the segregation effect. Okay, and this is the characteristic features of this problem, as well as all the mathematical details, have been recently reviewed by Korolev, Nelson, and other people in this paper in Review of Modern Physics. Now, as I said, you can either solve the problem by looking at the particle dynamics, or you can try to solve it by looking at the partial stochastic differential equation. I'm here now without the velocity, okay? So all the things I'm saying is without the velocity. There is a problem in solving this equation. I mean, there are a huge amount of literature trying to figure out each other's best method to solve this equation, and the problem is still not completely clarified. Anyway, in order to be as sure as possible, what we did was to solve this equation both with particle and in the continuous case, and compare the results in order to figure out whether the solution, the numbers that we get, are reasonable or not. So let's go back now to this case. You see that without velocity there is a segregation, and eventually, on a time scale which is very long, you may have global fixation. Now, when you introduce turbulence, you have sinks. So the particle goes immediately to the sinks, and then they fight in the sinks, and therefore you are killing part of the segregation effect, and you are reducing the global fixation time. Now, this is true when the turbulence is very strong. What happens when the turbulence is very small? So we perform a series of numerical simulations, and we found something which is non-trivial from our point of view. This is the expectation fixation time, so this is the expected global fixation time, assuming that everything is mixed in one box. So you are in one dimension. Suppose that the turbulence is reducing the space dimensionality in one sink. So you are able to say, okay, in one sink I can compute analytically what is the global fixation time, and this is what you get. The global fixation time that you have without flow is out of the blackboard. It's about 20 times bigger. And this is what you get if you look at the global fixation time as a function of the carrying capacity. You remember that I was pointing you before that even with very small compressibility, the carrying capacity can be reduced by a factor of 20%, so here. And you see that even when the carrying capacity is relatively large, the global fixation time is extremely small. So that even a small amount of compressibility can introduce a shrinking of the global fixation time. So if the mutation would be successful with much higher probability. And this is something that is a prediction that is now tested experimentally in an experiment in Eindhoven to see whether this is true or not. I mean, this is a non-trivial effect because it can explain how it happens that you have such a successful rate of mutation in the ocean with respect to estimates which are not completely in agreement with what you observe in the real case. So even a small compressibility effect can have a dramatic effect on the fixation probability, on the time scale of the probability of the global fixation in the system. Now the explanation for that is relatively simple and goes back to the previous slides. You see that when you have segregation, you have these boundaries. Now, if you look at the dynamics of these boundaries, you observe that the boundaries are doing a kind of random walk. Okay? So eventually, when two boundaries of the same species collapse in one point, the species die. But when you have a flow, the boundaries have random walk, and then you have a drift. Okay, so they go towards this, the point of sinks for a time scale which is of the order of one of the gradients of the sinks. And therefore, the characteristic time scale of fixation becomes the one determined by the flow, and not by the random walk. And if you put this idea and try to fit the observed fixation rate as a function of this characteristic time, which here is called f, you see that more or less you have a reasonable behavior, a reasonable fit between what you observe and what is the value of the shrinking effect. So this is more or less what we are now. It's just the beginning of the project, where we still have to understand whether some of these features are true or not in two dimensions, whether or not with inertial particle we will recover the same effect, and then eventually when we put all these things together, we can move to understand whether in the ocean something similar to what we are discussing is observed or not. The other point that I want to mention is that what is difficult in this field is to provide a very clear framework for experiment to be done. For instance, this kind of prediction can be done experimentally in microchannel by taking a very long channel, not trivial, because you have to put the bacteria in such a way that they must live, otherwise if they die, they have not food, you cannot make any experiment. And then you can put a sink, okay, in this channel, and then see whether or not the global fixation change as a function of the sink of the velocity. For the turbulent case, we still do not have any experimental idea how to make such a test, but if we are successful to show that this prediction is correct, as we think, then probably we will get some we can make a new effort to provide some experiment even for the children. And that's it. Thank you.
In your estimates, and also you explicitly say that more or less a case when I have underwater turbulence and that make a surface flow compressible. Now when you show this picture of ocean on the scales of tens of or even hundreds of kilometers, I mean, this is probably not the case, and I wonder what would be the source of compressibility of this flow on this scale.
The compressibility of, I mean, depends on the scale. I mean,
Tens of kilometers,
it's not
you're
a
wondering.
trivial question, and the answer is not trivial as well. If you look at the scale of few kilometers, then the compressibility is the one that you can have on the horizontal scale due to what you call frontogenesis phenomenon.
What?
Frontogenesis.
What is this?
Frontogenesis is a phenomenon that you have even in the atmosphere when you have sharp gradients of temperature which produce compressible effect in the system, in the vertical direction. If you have a bacteria...
You mean sinking of
Yeah,
of a cold water
Right.
and raising of...
Right. That time scale is of order of 8 hours, 12 hours, depends on the strength of the phenomenon. This is a large time scale.
relatively rare.
Relatively large.
Yeah.
On that time scale, you have two effects. First of all, that the growing rate of the bacteria is more or less the same. It's about 1 over 12 hours. And so it's in within the range of effect that you would expect. The second thing is that you have something which is going down here and something which is going up somewhere else. And the food is coming. I mean, the food that I am considering uniform now, it is not uniform in the ocean. No? So the two effects are, you should consider both effects. Okay? Now, at this stage, I'm not able to say what I'm observing is in one-to-one correspondence to what is observed in the ocean. But there are hints that if you do not introduce compressibility effect into the dynamics of the ocean, what you get is something which is very important.
That's why I'm asking, because there are hundreds literally of people who ask themselves and wrote papers, what are the social compressibility of a 10 kilometer or 50 kilometer scale?
Yeah.
And it's a huge body of work, and it's kind of different thing.
Yeah.
And of course, this ups very not they happen, but they're relatively rare effects, and
Well,
in a
they're-
sense,
they're
and
rare.
they usually happen in a very particular points. I mean,
Yeah,
that's
but they're
usually-
rare because they are not really very well observed. Remember that our observation in the ocean is extremely sketchy. I mean,
Well, that's because we know that the underwater turbulence is relatively rare. It's stable
I was
stratification mostly.
able to observe one of these effects about 30 years ago. Huh?
You were trapped at
No, it was
almost
a
drowned.
vortex forming in the Ligurian Sea, and the size was about 3 kilometers.
But that was
And we
30 years
observed
ago.
it. Huh?
That was 30
That's
years
what I
ago.
said. It's pretty clear. Yeah, well,
You are
good
doing
and
it
better life.
here.
No, but he is in Ligurian Sea only once every 30 years.
The problem is that I was observing it because I was there. So that phenomenon is occurring every year in a sketchy way all over the ocean. And we have no monitoring system to observe this effect, as we have in the atmosphere. So when you say it's relatively rare, it means that you are being relatively lucky to observe it, which is the other way around.
And lucky to survive.
No, well, it's not dangerous
Not
because
dangerous.
the vertical velocity usually in the ocean is sort of 20 or 20 meters per day. During this effect, the vertical velocity can become half a meter per day.
When you're 5
But
meters
you
down,
don't
you're
go
not
down.
there.
You can survive. Okay. This is one thing. The other thing is that the phytoplankton goes from 10 to the minus 6 centimeter up to 3 centimeter. Okay? So there is an effect of the size of these animals which should be taken into account and give you inertial effect. Now, we are not thinking of having a compressibility order 1. What we would like to understand is what happens when you have a compressibility small, but not so small to be negligible. And we see that even a small effect is not trivial in the system.
Yes, but what I'm saying is that if you can have compressibility due to potential part of the flow, but they are waves, and they behave completely different.
Yeah.
For example, a former student of mine, Maria, was to build a series of waves and currents together give this type of effect. But that would be completely different estimate, for example, correlation time in Lagrangian dynamics, because waves run and currents don't run, currents move locally. So it's kind of non-trivial what the real surface
Also, there
flow.
is
But it's
this
compressible,
work
sure.
done by Tell and other people that show that something similar, but there's nothing to do with the compressibility happens in open flows. Okay? And this has mostly to do with the Lagrangian chaos. Yeah.
I had the same question.
Okay, sorry, you get the same answer.
Same answer.
Well, sometimes you can get different answers.
Okay.
Okay. So, is there another question?